genetic variability, how we measure it in populations, and how we can construct some quite simple models which help us to understand it. I understand there's been a problem with the textbook. There are very few copies available. I apologize for that. There seems to have been some misunderstanding with the publisher. Hopefully that'll get fixed at some point. But the background reading is, optional anyway. It's up to you. And I'm giving references to papers which might be relevant. Again, that's optional on the slides for the lectures. And there's a few handouts which should have been made available to you with some of the more boring algebraic details, which I'm going to omit from the lectures. And it's kind of, again, up to you how much use you want to make of that material. So let's get started. So I'm going to talk about DNA sequence variability mainly, but we'll have a short introduction going over some of the material that you've already heard. So I'm going to go over this fairly quickly. So the first question, obviously, is why should we be interested in genetic variability? And I think Darwin historically was the first person to realize that what evolution does is to take genetic variability which is present within a population or within a species and use that to cause differences over time or over space between populations, and that's what we think of as evolution. We're thinking of a single species evolving at some time in the future. Its genetic composition will be different from what it is now, and in order to do that, you have to have genetic variability for the various evolutionary processes to work on. So it's a really fundamental problem in evolutionary biology is to understand how much variability there is and what sort of evolutionary factors or genetic factors are involved in causing that variability. There's also practical reasons for being interested in genetic variability, and that's becoming increasingly recognized even among people like molecular biologists. The animal and plant breeders, of course, for millennia, as somebody pointed out the other day, have been practicing selection for improved traits of interest to humans, and that, of course, requires the same kind of variability that evolution acts on. And Darwin, of course, in the first chapter of The Origin of Species, spends a lot of time talking about evidence from the study of domesticated animals and plants. Also, if you're a medical person, people are increasingly interested in genetic factors predisposing to disease, especially when you get very old, like me. Most of the things which go wrong with you are probably under some kind of genetic influence, and maybe 100 years after I'm dead, somebody will find out ways of curing whatever it was that killed me. So you may be in a better position than me. So people are making enormous use of studies of associations between variants and traits of interest, both in the medical area and in animal and plant breeding. And some of the stuff I'll be talking about, DNA sequence variation, is actually being used in a practical way in these areas. But I'm not going to discuss the uses today. So here's a discussion, here's the structure of the lecture. First of all, I'll just go over rather quickly some of the classical evidence that you've already heard about yesterday. Then I'll talk about how we can measure variation at the DNA level, and finally, how we can understand where that variation is coming from. So let's jump into number one. If you do have any questions, by the way, don't hesitate to stick your hand up and interrupt me. If I'm saying something which is incomprehensible or illogical, please point it out. So, classical genetics, which was done when I was young, where you get information by crossing different strains of animals or plants and looking at what happens, reveals the existence of what we call genetic polymorphisms in some cases. That is, distinct, discrete types which are reasonably frequent in the population. I'm not talking here about rare traits which are caused by obviously deleterious mutations, something like hemophilia, which are confined to one individual in several thousand. I'm talking about things which are relatively frequent. Like in European populations, there's a good deal of variation for eye color, blue eyes versus brown eyes. So that's an example of a discrete genetic polymorphism. You have two or more rather well-defined traits which differ and they're variable in the population. Biologists have made quite a lot of use of those in studying evolutionary problems, as we'll see. There are also more subtle polymorphisms, and one of the more interesting ones, which has historically been of great use to people working on fruit flies, Drosophila, like I have done for much of my career. These are chromosomal rearrangements in which a piece of DNA is taken out of the chromosome and rearranged. So here we have some kind of set of genes in the normal sequence. An inversion basically is a break and a rearrangement. So this becomes CB, and everything in between is also in the wrong order, and then outside the breakpoints of the inversion, you go back to the normal sequence. So that's a kind of polymorphism which is quite noticeable in some populations of the fruit fly Drosophila. And huge numbers of papers have been published on this. Most biologists thought this was terribly boring and irrelevant until it was discovered quite recently, using DNA sequence polymorphism, that actually human populations have examples of inversion polymorphisms. And then, of course, it becomes very exciting because it's humans, not flies. So here's an example of the human polymorphism on chromosome 17. Here's a set of DNA markers in one order on this chromosome, and you can see they're going the other way on the alternative arrangement. So one of these arrangements, I forget which, is in common with chimpanzees. This is what the other one is clearly the derived sequence. This is what was done in Drosophila. This is the classic species studied in the 1930s by Sturtevant and Dobzhansky. And here you have a giant chromosome from the salivary gland of the Drosophila larva. And this individual is carrying the arrowhead arrangement and the normal arrangement. What's happening here is the two are trying to pair up. So you get one sequence going in one direction here, and the other sequence going in the other direction here. So you get this characteristic loop where the chromosomes from the two parents are trying to maximize their pairing. So it's very easy in Drosophila, well, it's not that easy, but you can do it, to score the presence of these inversion polymorphisms in the population. And if you're really good at it, you can actually just read the order of bands. Characteristic bands on the chromosome are the wrong way around in the inverted and the normal sequence. So inversion polymorphism was documented. This is an example of a discrete polymorphism. This is the classic European land snail, Sepia nemoralis. And you can see this is actually Deborah's hand. This is a collection of snails outside a vineyard in France. I was inside with Deborah's father buying some wine, and she was outside collecting snails. Yellow, and here's a pink or brown form, and then you have banded ones, or non-banded, and different numbers of bands. So there's variation for the color of the snail shell, and there's also variation for the bands, or presence or absence of bands, and numbers of bands. And these are actually under quite simple single locus control, one for the color and one for the bands, and there's a few other polymorphisms which I won't bother with. So this is a classic example of polymorphism of a discrete nature. You're either this color or you're that color. You either have bands or you don't have bands, and so on. But there are relatively few examples of these. Most populations don't have a lot of traits which show these discrete classes. Much more commonly, we have what's called quantitative variation, particularly in what we call metric traits, ones which are continuously variable, like height, for example. So human height or body weight is an excellent example of a quantitative trait. They show normally quite a lot of variation. This is conveniently measured by something called the coefficient of variation. That's the ratio of the standard deviation to the mean. We heard about variance and standard deviation a couple of lectures ago, so I won't go into their definition, but that's just a measure of variability. You can scale it relative to the mean value of the trait, so you get a dimensionless quantity, and typically for these kind of morphological traits of organisms, the ratio is characteristically between 5 or 10%. So almost any quantitative trait you care to name shows variation at the phenotypic level. And that's an important distinction which has already been mentioned. I'll just remind you, the phenotype, for those of you who are not biologists, is what we look at or measure. Question is, does the variation in the phenotype have a genetic component? I'm doing the wrong thing here. So here's an example of a distribution of a quantitative trait, perhaps not terribly clear on the projector here. This is human height, so these are small individuals and these are big individuals, and the numbers of people standing in these different bits of the field represent the frequencies at which you observe relatively few small individuals, relatively few very big individuals, and quite a lot of individuals in the mean. The scale here is the standard deviation normalized, so the mean of the trait is 0, so we're -1 standard deviation below the mean here, 1 standard deviation above the mean here, 2 standard deviations here, -2 here. So about 2.5% of the distribution lies here, and about 2.5% of the distribution lies there. So the extremes occupy about 5% of the total distribution. So that's very, very typical of a quantitative trait. Question is there any genetic basis to that? And as you heard on Saturday, the answer is generally there is. I'm not going to go into the details. You've heard a bit about that already. If we measure resemblances between relatives, correlations between, say, parents and offspring, you can estimate the proportion of the variability in traits that's due to genetic factors. I haven't got time to go into the mathematical details of how you do this. It goes back to Fisher in 1918 and Sewell Wright at about the same time, and a man called Weinberg actually also independently developed the same methodology. But take it for granted that I'm telling the truth here. I may make mistakes, but I generally try and tell the truth as far as I know it. But 20 to 80% of the variance in traits, depending on the trait, is typically due to such genetic factors. More strictly, as you heard about variation in additive genetic traits, the ones which are handed down from parent to offspring. So this type of variation, of course, is tremendously important. Most of the stuff which goes on in agriculture are quantitative traits. Things like yield of crops, yield of milk in cattle, and so on. And also a lot of human genetic diseases like heart disease, blood pressure, for example, is a quantitative trait. If you have very high blood pressure, that's a serious medical problem. And it's continuously distributed and has substantial genetic component to it. But of course, just partitioning the variation into genetic and non-genetic doesn't really tell us anything about what's going on at the genetic level. We can't tell how many genes are involved in controlling the traits, what the frequencies of the alternative variants, the alleles of these loci are, and so on. So it's a sort of black box, as we've heard. We can say quite a lot about how a population will respond to the pressure of selection if it's got if we know how much variation in the trait is genetic, additive genetic, but we don't know anything about the details of the genetics. So there are limitations there. But one should bear in mind a very important point in the history of evolutionary biology is that knowing that genetic factors underlying quantitative traits really are the same as those of normal Mendelian traits is terribly important. I can't go into the experimental evidence for this due to lack of time, but take my word for it, really the only difference between quantitative traits and a discrete trait like the shell-color polymorphism is that the quantitative traits first of all have effects of the non-genetic factors affect their expression quite importantly, and secondly, the genetic components are not due just to single genetic locus with two variants at some known place in the chromosome. There's many, many different places all over the genome that affect the trait, and their individual effects on the traits are quite small. So you can't easily track the effects of individual genetic variants. But we do know for sure that there's nothing special about the genetic variants underlying quantitative traits. That was historically a big step forward in the early history of evolutionary genetics and the beginnings of the 20th century, because that means essentially we can think about evolution really in terms of changes in the frequencies of these alternative variants. even if we're thinking about a trait that's rather complicated and controlled by a vast number of loci. Now, there are, of course, always some exceptions. Not all variants are at loci on the chromosomes and inherited in a straight Mendelian way. Particularly, there are maternally transmitted traits in humans controlled by loci in the mitochondria. That's just a little genome of about 70 genes, 17 genes, which lurks inside our cell. It's derived ultimately from a bacterium. It's a very stripped-down bacterial genome, and it's more or less universally transmitted from mother to offspring. So although males receive a mitochondria, they don't transmit it to their offspring. And in plants, there's also the chloroplast. That's frequently also maternally transmitted, although there's some rather eccentric plants where it's transmitted through the pollen for reasons we don't know. Okay, they're not strictly Mendelian, but basically they still have the same DNA and the same stuff goes on, and it's really an unimportant exception. It's just the mechanics of transmission are different from normal Mendelian loci because it's from mother to offspring rather than getting equal contributions from male and female. So that means I'm going to really assume, for the purposes of this talk, that we're dealing with diploid organisms with Mendelian inheritance. You can also discuss haploid organisms like bacteria in much the same way. It's actually simpler because we don't have to worry about one set of genetic information coming from the mother and one set of genetic information coming from the father. It's more like the mitochondrial situation in which there's just one parent you need to consider, and you just have straight transmission between parent and offspring. So it's actually easier to deal with. So I'm not going to talk about it. We need to deal with the more complicated case, which is the rule for animals and plants. So we had this problem. We know that everything is Mendelian loci. It's located on chromosomes with a few exceptions. There's variation at these sites in the genome, causing the variation in the traits we're interested in, but we don't know how to measure the variation at those individual genes. And you had a nice talk about the use of gel electrophoresis yesterday, which was the sort of classic way of dealing with this problem invented in the 1960s. I was actually a postdoc with Richard Lewontin about the time some of that work was going on. But it's still a useful method, but of course it's been superseded, as was already said, by the fact we can sequence DNA. And if you're a geneticist, that's the genetic information. There's nowhere else to go. Once you have the DNA sequence information, that's all you're ever going to get. It's never going to get better than that. So we can use the fact that genetic information is encoded in the DNA, and we can answer the question of how much genetic variability is involved. That was question A on a slide or two back. Question B on that slide was what's involved in controlling that variation? What are the causes of that variability? Now, we really have the answer to question A, essentially. I mean, we haven't done it for every organism on the planet, but the general principles are absolutely clear, and I'll be showing you what those are. Question B, it's in some ways a more interesting question. What's controlling that variation is still an ongoing area of research. So there's still a lot to do, despite the fact we have an answer to question A. And so these methods for studying variability at the DNA level really started to get going in the late 1970s, where people invented methods for cloning pieces of DNA. That's taking a bit of DNA from an organism like us and inserting it into a bacterial cell, replicating it, and then you could get your hands on that bit of DNA and make a sequence of it. And you can study variation then in coding sequences, those bits of DNA which correspond to the structure of a protein or a functionally important RNA molecule. So that's an important distinction. Coding. So here we have a bit of DNA which goes straight into the sequence of a protein, or possibly, in some cases, a useful RNA molecule like ribosomal RNA. And then there's non-coding DNA, and that does not get into the protein or the RNA. And there's sort of two main categories of non-coding DNA. For those of you who are not biologists, the so-called introns, and then there is the intergenic DNA. So what's an intron? Anybody know? What is an intron? Sorry? Right. It's a bit of the DNA which is encoded in the same piece of the genome where the protein is being made, but actually it never gets into the protein. There's an intermediate, the so-called messenger RNA, and before you make the messenger RNA, you make an RNA molecule which contains the intron. There's a chunk of DNA. It's cut out. We don't need to know about the molecular biology of this, so it never gets into the messenger RNA, and so it never gets into the protein. And actually, in human genomes, a lot of what's inside our genes, those pieces of DNA which actually correspond to a protein, do not get into the messenger RNA. It's mostly introns. I'll show you a picture of that later. So that's one important category. So inside the coding sequence are actually these introns. And it's an interesting question why the hell they're there, and we don't really actually know the answer to that. But it's useful material when you're studying genetic variability, because quite a lot of the DNA sequence is introns, and in between the genes coding for proteins, there's also vast stretches, in the case of humans, of intergenic DNA. So that's also non-coding, not necessarily non-functional. I'm not saying these things don't contain sequences which are important, they clearly do, but not often as important as the amino acid sequence of the protein. So, coding sequences, sorry? It's more, well, come on to that, don't worry. So about 98% of the human genome is actually non-coding. Drosophila is a more respectable, clean organism. About 75% of its genome is non-coding. If you go to yeast or E. coli, which have even more compact genomes, much, much less is non-coding. So there's a great deal of variability among different organisms in the structures of their genomes. We're carrying around a huge amount of DNA we could probably do quite well without. Again, an interesting question why that is. So, getting back to the history of DNA sequencing, people invented this clever technique in the 1990s called polymerase chain reaction, which allowed you to amplify DNA from a very small sample and do it very specifically. And then they also had automatic sequencing machines. The classic literature on DNA sequence variation uses this method where you take a known region of the genome, PCR up the DNA from it, and then sequencing it using Sanger sequencing. Fred Sanger, who died recently, invented the most commonly used classic method of DNA sequencing. But now we have so-called next-generation sequencing. I don't know why it's called next generation, because everybody's using it. It's actually this generation, but there you are. And again, I'm not going to go into the technology largely because I don't understand anything about it myself. It's there. You can actually now sequence, in principle, entire genomes from multiple individuals of a species without any need to clone specific regions of the genome. So there are various papers describing studies of variability. The so-called 1,000 Genomes Project, which does it for humans, published a few years ago in Nature. There's now a 100,000 Genomes Project on hand in Britain, mainly for medical reasons. There's also a study by Chuck Langley, who was the leader in the study of genetic variability at the DNA sequence level using Drosophila melanogaster, the classic fruit fly, that was published recently in Genetics. I left the date out, it's 2012. So those papers are available if you're interested. The Langley one is a real stinker to read. It's about 100 pages long. So pioneering work actually was done by a guy called Marty Kreitman, a student of Dick Lewontin at Harvard in the 1980s. In those days, you did it all by hand. You got the DNA, which had been cloned, blown up, so you had bucketfuls of the DNA from a given strain of fruit flies. You had these huge gels. You ran the stuff out, and then you spent hours deciphering what was going on the gels. And it was an entire PhD, basically, to study 11 independent copies of a particular locus, the ADH, alcohol dehydrogenase gene, a very useful gene both for humans and fruit flies, because it breaks down alcohol. And he sequenced 2,379 bases of the DNA from each of these alleles. And then he looked, this was really the first real study of DNA sequence variation at the actual sequence level. There are earlier indirect approaches using restriction enzymes, but this was really the first DNA sequencing project. So most of the variants are what we call single nucleotide polymorphisms, SNPs. Now, for a visitor to Scotland, this is very confusing because you see signs all over the place saying SNP. It stands for the Scottish National Party, which is run by these deluded individuals who want to make Scotland independent of Britain. And I'll be, if that happens, Deborah and I will be put on a freight train and sent south. Anyway, but they're single nucleotide polymorphisms. In other words, they're just alternative variants at the same site in the DNA. I'll show you some examples of those in a moment. You already heard a little bit about that yesterday. So that's actually the vast bulk of variability in any organism, a single nucleotide polymorphism. And if you read the literature, people talk all the time about SNPs or SNPs. So what did Marty actually do? Well, actually he's discovered everything really we know about DNA sequence variability. Pretty much the same thing comes out again and again. So he showed there's a high level of variability at individual nucleotide sites. And most of this variability involves what we call silent changes, ones which don't affect the protein sequence of the ADH protein. The changes are either in the non-coding sequences or involve, as you heard yesterday, synonymous changes in codons, the little triplets that correspond to each amino acid. In other words, they don't change the amino acid sequence of the protein coded for by the gene. So you'll be hearing through the rest of the course quite a lot about these types of change, silent changes, or changes in non-coding sequences, synonymous changes. So basically, silent changes have these two different flavors, the ones which don't occur in bits of the DNA which code for protein, and the others which do but don't change the protein sequence. So ADH actually has a classic protein sequence polymorphism. There are two different variants, which are called fast and slow because they run at different speeds on an electrophoretic gel, and they correspond to a single amino acid sequence. I think the person who first sequenced the ADH gene was a man called Richard Ambler at the University of Edinburgh, who died recently. The fact that most variability, there's only really that variability was detected again by Marty. He saw only that fast-slow difference, single amino acid difference. He didn't see any other non-synonymous changes. Non-synonymous mutations are ones which do affect the amino acid sequence. So it's another little bit of jargon that's important to remember if you're not familiar with it already. So the fact that we see so little variation, and you can do a calculation of how much you would expect, and it's far less than you'd expect, in non-synonymous mutations suggests very strongly that the mutations affecting the amino acid sequence of a protein are actually selectively disadvantageous for the most part, and then we don't see them because they're wiped out of the population by purifying selection. So most of the variation that we see in coding sequences, the bits of the DNA that correspond to proteins, involves synonymous variants. So in the case of Drosophila, it's about 85% of the variants that we see at the DNA sequence level with respect to coding sequences are synonymous. And therefore, they must have much smaller average effects on fitness than non-synonymous variants. Okay? So lots of variability, the silent mutations in non-coding sequence or synonymous variants, but very much smaller amounts of variability at the level of non-synonymous changes that affect the protein sequence. And, as I say, non-coding sequence variants actually in Drosophila show on average somewhat less variability than synonymous variation, which suggests, and there's quite good evidence for this now, that actually quite a lot of these non-coding sites do have some functional significance in, say, regulating gene activity, things like that. Sorry, I can't hear what you're saying. In it's very difficult to directly detect the effects of any of these mutations on fitness, with some rare exceptions. So we'll be talking about that later in the course. Trying to infer exactly how much effects on selection these things have is actually quite a hard problem. It's not completely solved by any means. So in most cases, we don't know. Actually, when you compare different species, you get pretty much the same pattern as already been mentioned. So this is another way that you can come at the so-called neutral theory. The fact that most variation in populations involves things which don't affect protein sequence, and there's very much less variability in things that do, suggests, but it doesn't prove, that most variation in evolution might be due to selectively neutral variants, ones which have no effect on fitness at all, so the different alleles just don't have any difference with respect to survival or reproductive success. Or they might be what's called merely neutral. They have such weak effects, and we'll come on to what we mean by that later, that their fate's really mainly controlled by the process of genetic drift, which we've already heard about. And genetic drift really just refers to the process of random changes in variance due to the fact that the population size is finite. And I'll come on to this a little bit later in the lecture, exactly how we can model that. So that's sort of broad picture. I haven't made this quantitative in any way. I've just given you some sort of qualitative feel for the sort of general pattern you get from DNA sequence variation. But if we're going to be real scientists, I think it was Lord Kelvin, the famous thermodynamicist, who said if you can't measure it, you're not doing science. That's probably going a bit far. Geology is a science, and they don't measure much in geology. We need to know how to measure DNA sequence variability. So let's have a look at how we do that. So where do we get the information? Well, we get the information, as I've already said, from DNA sequences, and this gives us what are called haplotypes. We've got one genome from our mother, one from our father. We're only going to look at one of those two genomes. We're going to choose one at random. We're not going to have any sex discrimination here. We'll just take a random sequence from one of our two parents. That gives us a haplotype for that bit of the genome. Now, actually, when we're thinking about diploid individuals, if you think about it for a minute, it's not necessarily easy to get the haplotype out of a diploid individual. You have to use quite a lot of trickery. I'm going to ignore that. It's quite a hard problem. There are ways of getting around that. In organisms like Drosophila, you can make highly inbred individuals who are totally homozygous for one haplotype. Humans, it's more difficult. You're not allowed to inbreed people for some reason. So let's assume we can solve the problem and we can get a haplotype. So what does a haplotype look like? It's a DNA sequence for that chunk of the genome. Here, this is a gene called glucose 6-phosphate dehydrogenase. It's a very interesting gene, for reasons I'll talk about later. As you can see, it's got some exons. These are little bits of DNA which correspond to protein, interrupted by long introns. So as I already said, a lot of the genome is introns. I'm only showing you here sites which are variable in the human population. So this number here corresponds to the location in the sort of arbitrary coordinates 215, base pair 215. It's AAA, and the chimpanzee here is different. So there's variation, but it's only between us and the chimpanzee. So we are different from chimpanzees. There's another site which is non-variable in this sample from the human population, but it's different from the chimpanzee. Here we have a variant, G, and the box here means that it's actually a non-synonymous variant. So we actually do have a non-synonymous variant in this exon. But notice we've gone from 215 to 278 with no variation either within the human population or from chimpanzee. So there are not many, actually, it's about 1% difference between us and the chimpanzee at the DNA sequence level. There's even less variability in the human population. Here we have some synonymous variants in the human population. But these numbers here give you some idea of how little variation there actually is. We're jumping nearly 50 base pairs, and there's no variation either between us and the chimpanzee or within the human population. And as you can see, Marty Crichton was right. Most of the variation is either in the intron sequence or it's in synonymous variants within the exons. And because there's so much, the introns are so big, it's mostly intronic in this case. This is all from one population, yes, yes. Actually, human population, yeah. Sorry, these are different variants within the human population, a single human population. Interestingly enough, most variation, this was pointed out by Richard Lewontin, and I think Mike Whitlock might talk a little bit about this later, there's not that much DNA sequence variation, differences between different human populations. 85% of the variability in the human genome is really represented within this room. Included, even if you take the Europeans out, actually at the DNA sequence level, we're almost the same as anybody else. The Indians are more or less the same as the Chinese and so on. So at the DNA sequence variation level, there's very little differentiation between human populations. There's enough that you can see it, but it's minor compared with the variation within populations. Oh, that's an aside, but yeah, it's a good question. This is all from one population. Yeah. No, okay. Yeah. Okay, let me... So the ones in black boxes here are the non-synonymous changes. Okay, these are where the amino acid sequence of the protein is changed. These ones here, if they're in the exon, are synonymous changes. These ones here are outside the exon. They're in the introns. So there's the non-coding differences. Okay. As you can see, most of the variation is in the non-coding sequence or is synonymous. There's relatively little non-synonymous differences. And in fact, none of the differences between us and chimpanzee in this example are non-synonymous. There's one question which might have already occurred to some of you. DNA, of course, is a double-stranded molecule. There are two strands in the DNA. Conventionally, you just get one sequence. Of course, the other sequence is automatically given to you by the rules of Watson-Crick base pairing, so A pairs with T and G pairs with C, so you don't bother with giving two. There's always a question of which of the two strands you're using, and there's a convention, and I never remember what the convention is. Deborah, do you remember? I think it's the one which actually corresponds to the sequence of the messenger RNA, but you have to be a bit careful about that. You can get rather odd results when you do your DNA sequencing surveys if you're not careful about which strand you're representing here. Computer's gone to sleep. Okay, so is that reasonably clear now? That's what the basic data is. And now, if you think about it, the human genome contains 1 billion 10 to the 9 base pairs. If we look at DNA sequence variation at the level of the entire genome, we have a massive amount of information. Yes?
So even though the introns are really long in this gene,
yes.
You observe roughly the same number of polymorphisms within the introns as you observe within the exons.
No, actually,
the number of
well,
changes scaling within
no. I think if you do the numbers, the variability at the site level is about the same in introns as synonymous sites.
So even though you'd expect a longer sequence of DNA to accumulate more,
No, it's about the same. If you do it on a I'll show you how to calculate it on a per site basis in a moment. It's a good question, but this is possibly a bit misleading, this slide. But in general, the level of variability is about the same for synonymous sites and intron sites.
Even though the length varies.
Yeah, per site. So if you add up over all sites, there's more variability in the introns because there's more sites. So we really have to have a way of normalizing the variation by the number of sites we're looking at. We'll see how to do that in a moment. I'm just being qualitative here, not quantitative. So in fact, in the human genome, there's probably maybe 10 million single nucleotide site variants in a sample of, say, 100 different genomes. I don't know what the exact number is, but people are using literally millions of SNPs in human genetic surveys of human genetic variation. So there's a huge amount of information there. We need to reduce this to something which we can manage. So that's what I'm going to talk about next. How can we reduce this huge amount of variation into a few simple numbers? Because you can't make sense of just long lists of haplotypes. That's not the way our minds work. So, how to measure DNA sequence variation? I'm assuming we've got independent haploid variants from the population. I'm going to call those alleles. So these are 3 different alleles, different haploid sequences. So allele 1, allele 2, and allele 3. You can see the red here indicates a different base pair. So A is G here. This G is A here. And here we have C, and here we have T. So we have 1, 2, 3, 4 nucleotide site variants, which we can call polymorphisms in that sample. There are 3 bases in this sequence. This is completely imaginary, but it doesn't matter. So one measure of variability is what's called the nucleotide site diversity. We take a pair of randomly chosen alleles and we can ask what's the frequency with which they differ at a given site. Let's see how we can calculate that from the data. What we do is we take all possible pairs of different sequences and determine the numbers of differences between them. So there's actually 3 possible pairwise comparisons here with 3 sequences. So we take total number of differences between all those possible pairs, and then we divide it by the number of sequences that are being compared. That's actually k times k minus 1 over 2, if they're k alleles. In this case, it's therefore 3 times 2 over 2, 3. And then we again divide as well by the number of bases that we study. So this comes back to that question. We can take the variability which we have for the whole sequence and then normalize it by the number of bases, because otherwise we can't compare things. So usually, if you're doing it right, you compare variability at the level of the individual nucleotide site. So let's just do the numbers here. I think I've given you an example for the one of the problems, so we don't need to belabor this point. K equals 3, k minus 1 over 2 is 3. The number of pairwise differences, is, if you go back to those sequences, you can check this for yourself, is 1 plus 3 plus 4, that's 8. So there's 8 pairwise differences. So our value of what's called pi, nothing to do with the ratio of the circumference to the radius of the circle. It's a silly symbol, because it does get confused with that. It's 8 divided by 3 times 30, 0.089. Okay? So we now got the probability per site, if you like, that 2 randomly chosen sequences differ. And that's a natural measure, I would say, of variability. It's very like the heterozygosity for enzyme loci that we were hearing about, except we're doing it now at the base pair level. So we've completely removed the number of bases. We have a standard of comparison which we can use for different species, different populations, different genes. There's another much cleverer way of doing it. This was invented by a man called Jeff Waterson, who's an Australian mathematician. We simply count the number of sites that are segregating in the sample. That's often called S sub k. K is the sample size. So we just count the number of polymorphic sites. And then we divide that by the number of bases in the sequence, and then as a magic number, which in population genetics is often called Waterson's correction factor. Kavita told me it's a well-known term in mathematics for this, which I've now already forgotten. Thank you. So we take this, it's 1 plus 1 over 2 plus 1 over 3 up to 1 over 1 minus the sample size. And then we get what's called Waterson's theta, or theta sub W. So if you read the population genetics literature, you'll see Waterson's theta or theta sub W, or sometimes people just call it theta. And it's this measure, number of segregating sites, divided by the number of bases in the sequence and by Waterson's factor. So let's do the sums here. And as we'll see, the populations of equilibrium and there's no selection. In fact, theta W's should be fairly similar to pi in its value. Mathematically, the expectation, the average over a large number of independent evolutionary replicates of theta is the same as the expectation of pi. So in this example, S sub k is 4, A sub k is 1 plus 0.5, because we've only got a sample size of 3, so it's 1.5. We divide 4 by 30 times 1.5, and we come out with 0.089. Exactly the same answer as for pi. Those of you who are mathematically astute will realize that, in fact, for 3 alleles, it's absolutely necessary that theta equals pi. It's constrained to be that way. But in general, for larger samples, there could be a difference between theta and pi. They don't have to come out exactly the same algebraically. In this case, they actually do. But in general, they're going to be different. And the extent of difference might indicate how bad the assumption of equilibrium under mutation and random factors actually is. So these are the 2 measures. You can use either. Theta, of course, makes assumption. It makes assumption that the variability is neutral and the population is in equilibrium, which may not be true, either of those. Well, the size of SK will get bigger. In fact, it's roughly equal to the logarithm of the sample size, but it goes up rather slowly with the sample size. So actually, you don't get a huge amount of extra information from very big samples. So from the population genetics point of view, it's utterly pointless to sequence 100,000 genomes. You could probably get almost as much information sequencing, say, 20. So we can estimate theta and pi. Here's some typical values. E. coli, silent sites, 0.02. Quite high, 2%. The highest one on record, I think, is about 15%. It was recently reported from a species of nematode. Drosophila is actually quite similar to E. coli. Humans are way down. We're one of the least genetically variable organisms at the DNA level. Pi is about 0.001. And as I already mentioned, but if you add up over all the different bases, instead of looking at a single base, you have millions of variants. And here's a picture pinched from our textbook of the distribution of pi values in different organisms. These are in percent. 8% at that time was the highest value that was reported. This was from some sea organism, the sea squirt, Cyona. And as you can see, here are humans down here. We're really very depauperate in terms of variability. So there's huge differences between different species. You can go to nearly 20% pi to 1 in 1,000. We'll maybe discuss why later on. Just to wrap up this descriptive part of the talk, there are other types of DNA sequence variation. For example, you can have insertions of what are called transposable elements. These are large chunks of DNA which can move around the genome and insert themselves in different places. These are usually found outside coding regions because they probably induce harmful mutations if they go into a coding sequence. Then we have small insertions or deletions. These are more abundant, so-called indels. That's short for insertion or deletion. These are usually a few bases in length, but sometimes can be much bigger. And they're usually also found outside coding regions because they can disrupt the sequence of the protein if they're in a coding sequence. So you don't usually see much of that when you align different coding sequences. And then there are the microsatellite loci, which were referred to yesterday. These are little arrays of repeated sequences, and the variability really consists in the repetition of the sequence. So you have different copy numbers in different individuals, and they've been, for that reason, very useful. So you might have one individual with CA and another individual with just CA. So that's what's called a microsatellite, short runs of repeated base pairs. Okay, now the last part of the lecture, where does all this variability come from? And that's where we're going to start to do some real population genetics in terms of making models to understand the process. So let's just start at the simplest place, where nothing is going on. There are no evolutionary forces acting on the population. Well, what do we mean by an evolutionary force? That's anything which would tend to perturb the current situation. So mutation, random changes in the DNA sequence are one such force. DNA sequence does not replicate perfectly. Sometimes errors are made. That's a mutation. Selection, we've heard quite a lot about, so I don't need to belabor that. Or you can have immigration of individuals into the population. And finally, if the population is so maybe small in size, so frequencies fluctuate randomly from one generation to the next. Right. The answer, what happens if nothing is happening, is that nothing changes. With at least thinking about variability at a single locus or a single nucleotide site. And that's opposite, as was mentioned in the lecture on Saturday, to what Darwin believed. He believed in something called blending inheritance, where the genetic material from the two parents is mixed together each generation. The result of that is actually variability. It's very quickly lost from the population, because if you have a big individual mates with a small individual and the offspring are in between, they blend, then when you have another pair of individuals, big and small, they produce intermediate individuals. The two intermediate individuals mate, they'll produce intermediate offspring, and all that variability has disappeared. In fact, it roughly halves every generation. So Darwin was really worried about this idea that variability was vanishing from the population, but because Mendelian inheritance is particular rather than blending, we don't have that problem. At a given site in the genome, the contribution from either parent is transmitted intact to the eggs or sperm, the gametes. And so Darwin actually was very worried about this supposed loss of variability, and he in fact appealed to direct effects of the environment on the organism, which were then transmitted to the next generation, so-called Lamarckian inheritance, to generate the variability which is being lost each generation. And historically, Fisher's famous book, The Genetical Theory of Natural Selection, because we now know that it's Mendelian inheritance, not blending inheritance, the problem is illusory, and Darwin's solution to it is wrong. There isn't much evidence that inheritance required characteristics. And if variability is present in a population, there's nothing in the Mendelian mechanism to make it go away. It's just going to sit there. So that we start with a situation where we don't have these three genotype frequencies, we get them in the next generation. And you can generalize that to an arbitrary number of different variants. We can stick to 2 for the sake of argument. That's what we'll be dealing with mostly in the course. So after one generation of random mating, and emphasizing mating's random, so we're not choosing mates with respect to their genotype, and it's a reasonable assumption. I don't think organisms go around with a DNA sequencing kit and decide on their mates on the basis of their status. That may come, of course. P squared 2pq q squared, a very famous result in population genetics. It's a completely trivial piece of algebra, of course. G.H. Hardy was very ashamed of it because he was a very, very pure mathematician, and he hated the idea that mathematics could actually be used for anything. This is probably his most famous result. Life is unkind sometimes. Anyway. But that doesn't tell us where the variation is coming from. That tells us that the Mendelian mechanism conserves variability, at least at the level of individual nucleotide sites. And you can extend this to non-random populations. They don't end up in highly Weinberg proportions, but they'll still have an equilibrium. Same result holds. Because basically, in meiosis, A1 does not talk to A2. They just go at random frequencies into either of two gametes, with some exceptions, of course. So let's look at the question of where the variants come from and what's caused them to get where they are now. Mutation. That's the ultimate source of all variation. If there was no errors in DNA replication, life would be very boring. We'd all be some kind of small molecule in an RNA soup. But we have to have variation. And that variation comes from changes in the genetic material that are transmitted from parent offspring. That's all we mean by mutation. We're now in the fortunate position, DNA technology, that we can now measure the number of mutations that arise each generation at the DNA sequence level. So this is a nice paper that was published in Nature a couple of years ago on humans. There's another quite nice paper that was published recently on Drosophila, where they've actually looked at the difference essentially between parent and offspring and counted up the number of new mutations. So we can estimate the mutation rate, as it's called. That's just the rate per site per generation at which new variants arise. So parent has a particular sequence. The offspring have a different sequence. The probability that they differ in humans is about 1 times 10 to the minus 8 over one generation. In Drosophila, it's somewhat less, more like 5 times 10 to the minus 9. Bacteria, it's a lot less. They have very short little lives. They just divide every generation, whereas we have several cell generations between organism generations. So it's not very surprising that the mutation rate here is higher than it is in bacteria. So these are very small numbers. The probability that a mutation happens is small. I'm giving you the numbers here for single nucleotide changes. There are other kinds of mutations, inversions, I mentioned those, indels, insertions and deletions. They all happen, but they happen at a lower rate. Most mutations are single nucleotide changes. No, they're not. No. That's in things like bacteria, but it doesn't apply to higher organisms. Yeah, no. It breaks down. It's called Drake's rule, but it doesn't apply to higher organisms. So mutation is not a very effective evolutionary force. It's important for spitting variation out. It comes into the population, but of course it's present in a single individual, the new mutation. It doesn't do much for the population as a whole. So it's not very effective at changing the composition of the population. So let's have a look. It's going to be the first population genetics model. What's the effect of mutation on the composition of a population, assuming nothing else is happening? So let's assume we've just got two alternative variants. So this might be a GC base pair versus an AT base pair. People are quite interested in the difference between GC and AT. So we're ignoring more complicated situations. Now, the standard model in most population genetics is what we call discrete generations. So that's going to come in time and time again. What does that mean? It means that individuals are born so you have new zygotes. So the zygote is the sperm fuses with the egg. That gives you the zygote. They're formed at the beginning of the generation. Then these individuals go through some period where they grow up into the adult individual. The adults then mate with the other individuals in the population. If it's a sexual population, then the whole thing goes round and the adults then die. So there's no overlap of generations like we have in human populations, where there's a continuous range of age of reproducing individuals. We're simplifying things by this discrete generation model. It's an interesting question, what happens with overlapping generations. I spent a good deal of my misspent youth studying this problem. It's mentioned briefly in the textbook, but the results are basically usually pretty much the same as with discrete generation models. And it's much easier to handle these. And these apply very well to something like an annual plant, where the seeds are formed in the summer, they sit in the earth till the next summer, next spring, and then the seeds hatch out, or whatever seeds do. There's some technical term in botany which I can't remember, and they grow up into adult plants, and the whole thing goes round again. So the mutation model is very simple. A1 can mutate to A2 at some rate u. That's the probability that a random copy of A1 in a random individual changes to A2. And we can have also reverse mutation. It's important to remember with single nucleotide variants that mutation can go in either direction. So you can mutate from GC to AT, or you can mutate from AT to GC. Then you can get the new frequency, which I'm going to call P prime. So there's a probability, the frequency of A2 is 1 minus P. There's a probability V that changes to A1. So the new frequency of A1 is the mutation rate from A2 to A1 multiplied by the frequency of A2 in the population. And the other change, it shouldn't, and then there's the probability it doesn't change to A2 multiplied by its own frequency. You can sit down afterwards and scratch your heads and check that this is correct. Hopefully it is. So you've got a new allele frequency, this simple linear combination of the two allele frequencies, 1 minus P and P with the two mutation rates. And you get the new gene frequency, and you'll see this notation again and again. The change in gene frequency, delta P, is just obtained by taking P away from P prime. And if you do a little bit of algebra, which I'm not going to do, you can get this result. Delta P is equal to V times 1 minus P minus UP. So as you might expect, the change in allele frequency is dependent on the pre-existing allele frequencies and the two mutation rates. So it's a very simple linear equation for the change in frequency of a variant under mutation pressure. So it shows basically, not very surprisingly, that the rate in change of allele frequencies is of the same order as the mutation rate. So in fact, if A1 is initially very rare in the population, so P is close to 0, if you look at that equation, you can see that A1 will start to spread at a rate of approximately V. That also shows there's an equilibrium. You can come to a situation, go back to the slide, where delta P is 0 because V times 1 minus P is equal to UP. So there's no change in allele frequency. That's an equilibrium. Nothing's happening. That's what we mean by an equilibrium. Delta P is 0. You can check for yourself, if you do a little bit of algebra, that the equilibrium frequency, and I'm going to use the notation star for equilibrium frequencies, is actually V over U plus V. And that's what gives you delta P equals 0. Interestingly also, this equilibrium is stable. What do we mean by that? It means that if you start below it, you'll go up, and if you start above it, you'll go down. So you approach it from either direction. Again, that's a concept which will come in again. Of course, it's a very familiar one in physics. But this is the simplest example, probably, of a stable equilibrium in population genetics. So what this is telling us is if we had a huge population with no random fluctuations in allele frequencies, and we had mutations backwards and forwards between these two variants, we would, in fact, have a stable equilibrium frequency. But actually, of course, the population is small and is finite in size, and we probably never get very close to this equilibrium, even if there's no selection. Yeah? Any questions? Right. So mutation pressure could, in principle, produce DNA sequence variants that are present at intermediate frequencies. But the time that it would take for this to happen is hundreds of millions of generations, okay? The mutation rate in humans is 10 to the minus 8. That means 10 to the 8, which is 100 million generations, it would take to get much of a change in allele frequency due to mutation pressure. 100 million generations in humans. The human generation time is about 25 years. So we're talking about, you know, a very long time, a billion years or so. So it's a very, very weak pressure. And that's an important conclusion. This is strongly emphasized by Wright-Fisher in the first chapter of the genetical theory. It's a book which is well worth reading if you've got lots of spare time and like scratching my head a lot, because it's not very easy. But the first chapter is nice and easy. It gets more and more difficult. And then it gets crazy towards the end. Other evolutionary forces, selection and genetic drift, are much more likely to be important in actually controlling the frequencies of the variants that we see. Mutation is producing the variants. It's the other forces which are causing them to spread in the population. So, this is the last part of the lecture. We're going to go rather quickly over some actually very simple concepts, but you may not feel that, that allow us to predict how much variation there is. And we're going to make the simplest possible assumption here, which we'll relax later on, that the variants are selectively neutral. And this is the assumption that underlies the neutral theory of molecular evolution, which you heard about yesterday. Even if you don't believe in the neutral theory, it's a very useful null hypothesis against which you can test data. So you can look at it in two ways. Moto Kimura thought it was the absolute truth. The rest of us think it's a useful working hypothesis against which you can use to reject it. So, neutrality is the case. Mutations can only rise to intermediate frequencies in a population, providing there's no immigration. So I'll be assuming here the population is what we call closed. There's nothing coming in from outside as a result of genetic drift. This is the process of random sampling of variants in a finite population. And this is a very important discovery in population genetics, that this random sampling of variants could be a significant factor. It has two important aspects. The population tends to become genetically uniform because there's an increasing tendency for all copies of the genes at a particular position in the genome to be descended from one single ancestral copy. We'll go into the maths of that in a moment. Also, of course, independent populations will tend to diverge because the whole process is random. So we'll be going one way in one population and another way in another population. And that's what gives us differences between populations and ultimately differences between species. And you can think of the whole process of genetic drift as being completely analogous to the tossing of a coin. So in the sort of classic ways of studying genetic drift, we have in one generation frequencies P and Q of A1 and A2. Then we imagine that we produce a large collection of gametes. So we have some organism which is producing an enormous number of eggs and sperm. We can imagine some kind of marine organism sitting there in the ocean. The time comes to breed. They produce large numbers of eggs and sperm. These drift around at random in the column, and then you make the next generation. And so if we have, say, n individuals here, we have 2n gene copies, right, because it's assuming a diploid organism. So these are the 2n parents. They're going to go back to 2n offspring in the next generation if the population size is constant. And each gene in an offspring can be regarded as a random draw from the gametes in the population. The frequencies in the gametes are P and Q. We then take 2n samples of genes from that pool of gametes. So there's going to be some small change in allele frequency, delta P, and Q, of course, will be minus delta P, because P plus Q has to equal 1, by the sampling process. So if the population size was, say, 5 and we had 10 copies of the gene, it's like tossing a coin. If the allele frequencies were 0.5, you don't expect to get exactly 5 of A1 and 5 of A2, that you might get 4 of A1 and 6 of A2, or you might get even 0 A1 and 10 A2 as a result of the random sampling. And this is the classic distribution in probability theory known as the binomial distribution. And this model, where you have this assumption we have basically 2n gene copies in the parents and 2n gene copies in the offspring, where the offspring generation is a random sample from the parent generation, is called the Wright-Fisher model. I would claim it should be the Fisher-Wright model, since F comes before W, but there it is. It's entrenched in the literature. And in fact, Fisher, in 1922, was the person who introduced this model. So that's the sort of basic model of genetic drift. Random sampling from one generation to the next, and see what happens. So here's an example of a sort of imaginary simulation. Here's the frequency of A1 over time. And we start off with something like 40% frequency. Here we have these little random changes. And sooner or later, you end up fixed. Let's do another trial. Suppose we've got an independent population starting at the same frequency. We get something different, because the process is purely random. There's no directionality to it. So in this case, we've ended up with evolution. Two populations are different. Another fixation here. Another fixation there. And as you can see, there's variability here maintained until you get loss or fixation. And under this model, loss or fixation is actually inevitable. Sooner or later, you're going to hit the boundary, 0 or 1. And basically, the probability that you hit either boundary is equal to the initial frequency in the population, because the process has no directionality. The average, the expected gene frequency, never changes. It just sits here. But you get some populations go this way and some populations go that way. And so you work it out. The probability that you end up fixed for this allele is equal to P. The probability you end up fixed for that allele is equal to Q, because there's no directionality. Okay, that's really just summarizing what I wrote up on the board. Now, there are many different ways of modeling genetic drift. I'm going to look at it in terms of identity by descent, which we've already introduced yesterday. Two alleles, the same site in the genome, are identical by descent if they trace their ancestry back to a single ancestral allele. Here's a cartoon of this. This is a closed population of two breeding individuals. Here's the founding generation. We're looking at a particular site in the genome, say a single nucleotide site, and we're going to label those as distinct. They may not be different in DNA sequence, but we are omniscient beings, and we will distinctly label this. We will separate out the maternal and paternal alleles at this site in the genome for these two individuals. We have random reproduction. This is actually what basically mating between full sibs here, brothers and sisters, to get the final generation. As you can see, by chance, all of these alleles present in generation 3 trace their ancestry back to this ancestral allele. So they're identical by descent from this common ancestor, right? That's got to happen if you go on inbreeding by sib mating. After providing there's no selection, sooner or later, all the alleles in the population will trace their ancestry back to some initial founder allele because of the finite size of the population. Okay? That's really all that I'm going to be talking about, is the mathematics of this process. What we're going to do is look at it backwards. We're going to take the current generation and ask questions about tracing the history of the alleles in the current generation back to this founding generation. But before we do that, let's just look at this. This is the Wright-Fisher. We're going to assume the Wright-Fisher model. I've already introduced that. What this means in practice from the Wright-Fisher model, there are no systematic differences in reproductive success among individuals. There's purely random contributions to the next generation. Actually, for a large population, do the math, the number of offspring per individual has to follow a